Good morning, everyone. I would like to explain you how we use gene editing to reduce gluten immunogenicity in wheat. So wheat grains contain gluten, which is actually a complex of different proteins that are coded by 5 large gene families. On one hand, you have the glutenins that give dough elasticity, and on the other hand, you have the gliadins that gives dough viscosity. And it's the ratio gliadins over glutenins that give the bread dough quality. And gluten is present in most of the edible products, either as a main compound, such as in bread, or as a hidden compound, such as in processed food, where it's used for its viscoelastic properties. And that can actually be a problem because some gluten protein fragments that we call epitope can trigger celiac disease, which is an immune reaction that provokes intestine disorder to 1 to 2% of the population who have genetic predisposition. And those immunogenic epitopes, they are mainly carried by the gliadins, while the glutenins that are the most important for bread quality are usually safe for celiac disease. And in terms of remedies, so far, no drugs have been commercialized for long-term treatment. They are only for emergency. Therefore, gluten-free diet is the only solution at the moment, but it's very difficult to follow since gluten is everywhere. And currently, the gluten-free substitute product contain a lot of additives to compensate for the lack of viscoelasticity, and those additives are unhealthy on the long term. But how does actually gluten trigger celiac disease? So the immunogenic epitope of the gluten in the intestine, they can go through the epithelial barrier and they will get in contact with the cells from the immune system. And in case of genetically predisposed people, those immune cells, they will have receptors that are going to bind the epitopes that are immunogenic. And this is going to trigger the autoimmune reaction leading to celiac disease. However, it exists some variants of those epitopes that have slightly different protein sequences, and those are no longer binding to the receptors and therefore not triggering celiac disease. So based on this knowledge, we decided to use CRISPR-Cas9 in order to target the immunogenic epitopes from the gliadins in order to generate wheat with hypoimmunogenic gluten, where other conventional breeding technology didn't work out because of the number of gene copies and the complexity of which is hexaploid plant. So the idea is to create mutation in the immunogenic epitope to render them non-immunogenic, or to simply knock out the gene to avoid the protein to be expressed, or since the gluten genes are next to each other, to remove complete clusters of gluten genes in order to decrease overall the gluten immunogenicity without touching the glutenins that are important for bread dough quality. So how far did we get? Well, the variety of wheat we started with had 87 alpha-gliadin genes, and we succeeded to mutate or remove 1/3 of them. And looking further at the number of alpha-gliadin sequences that correspond to the different alpha-gliadin gene copies, we could realize that we remove a full fragment of the DNA that contain alpha-gliadin clusters. And looking further at the profile of the expression of those proteins on the acid page, where each band shows a different gliadin protein, we can see a clear reduction or absence of expression of some of the gliadin protein. And further than that, the plant needs to be analyzed for less immunogenicity. And another group in Spain used exactly the same approach and could prove that there was a decrease by 85% of the immunogenicity of the line that they edited. However, despite this great scientific progress, the availability of such a hypoimmunogenic wheat would be impacted by gene editing regulation, because in countries where gene editing is not regulated as GM, this technology is going to enable our hypoimmunogenic gluten wheat to be easily, cheaply, and fast commercialized, and it can be grown everywhere. However, in countries where gene editing is regulated as GM, such as in Europe, the same hypoimmunogenic wheat would have to follow the expensive and lengthy GM rules, and therefore the area of growth of this plant is going to be very restricted. In addition to that, imported editing plants would be a problem because they are not labeled and they are not tested for GM risk assessment, so they shouldn't be allowed to enter the European continent. So European celiac patients will probably not be able to eat bread with safer gluten anytime soon, while American people are likely to do so. And on that remark, I would like to thank you for your attention.